Inside laboratories around the world, scientists are keeping human cells alive using a strange golden liquid: skin cells, muscle cells, nerve cells, immune cells. Without this liquid, many of the experiments that power modern medicine would simply fail. Cancer research, vaccine development, drug testing, stem cell science, all of them depend on something called Fetal Bovine Serum, or FBS. But what exactly is it? Why do scientists rely on it so heavily? And why has it become one of the most controversial ingredients in modern biology? To understand FBS, we first need to understand a simple problem. Human cells are surprisingly fragile. Inside your body, every cell lives in a carefully controlled environment. They're constantly supplied with nutrients, proteins, hormones, minerals, and thousands of chemical signals. But the moment a cell leaves the body, everything changes. The support system disappears, the nutrients stop arriving, the signals fade away, and without help, most cells begin to die. Scientists needed a way to recreate the cell's natural environment inside a laboratory. And that's where FBS enters the story. Cells aren't just tiny blobs floating around. They're living systems constantly consuming resources. They need glucose for energy, proteins for building and repair, hormones for communication, minerals for countless chemical reactions, and perhaps most importantly, growth factors. These ingredients tell cells when to grow, when to divide, when to repair themselves, and when to survive. Without these signals, many cells simply stop functioning. Scientists needed a biological soup capable of providing all of these ingredients at once, and nature had already created one. Serum is the liquid portion of blood left behind after blood cells and clotting proteins are removed. Think of it as a nutrient-rich cocktail. It contains proteins, hormones, growth factors, vitamins, and countless molecules circulating through the bloodstream. For decades, scientists discovered that serum was remarkably effective at supporting cells grown outside the body. But not all serum is equally useful. One type turned out to be especially powerful: fetal bovine serum. FBS contains an extraordinary mixture of biological ingredients, growth factors, proteins, nutrients, hormones, attachment molecules, and many other compounds scientists are still studying today. You can think of growth factors as biological text messages. These molecules attach to receptors on cells and tell them what to do: grow, divide, repair, damage, stay alive. Without these signals, cells often stop growing and eventually die. With them, they thrive. This is why FBS became the gold standard of cell culture. It simply works, and it works for many different types of cells. The impact of FBS on science is difficult to overstate. Researchers use it to grow cancer cells in laboratories, to study stem cells, to test potential medicines, to develop vaccines, and to better understand how the human body works. Many medical discoveries over the past several decades have relied on cells grown using FBS. Without it, progress would have been much slower. But this is where the story takes a complicated turn, because FBS has a controversial origin. Fetal bovine serum comes from bovine fetuses obtained during the cattle production process. Scientists discovered that serum from fetal blood contains exceptionally high concentrations of growth-promoting molecules. These growth factors make it particularly effective for supporting cells in the laboratory. For researchers, FBS became one of the most reliable tools available. But for many people, its origin raises difficult ethical questions, questions that science continues to debate today. This is where biology stops being purely scientific and becomes philosophical. On one side, FBS has contributed to countless scientific advances: cancer research, drug development, vaccines, stem cell therapies. On the other side, concerns about animal welfare continue to grow. Is there a better way? Can science achieve the same results without relying on FBS? And if alternatives exist, why aren't we already using them everywhere? These are not easy questions, but they are important ones. Scientists around the world are actively working on replacements. Some laboratories use serum-free media, others rely on synthetic nutrients. Researchers are developing purified growth factors that can replace some of the signals provided by FBS. The goal is simple: create a system that is safer, more consistent, more ethical, and easier to reproduce. Progress is happening, but replacing something biology evolved over millions of years turns out to be incredibly difficult. Biology rarely gives us simple answers. Living systems are incredibly complex. Change one molecule, and countless other processes can be affected. A substitute that works perfectly for one cell type may fail completely for another. This is why replacing FBS isn't as simple as swapping one ingredient for another. Every cell dot, every experiment, every biological pathway introduces new variables, and that complexity slows progress. The story of FBS reflects a larger truth about science. Every discovery comes with trade-offs. Every breakthrough forces us to ask difficult questions. Science gives us powerful tools, but society must decide how those tools should be used. Understanding the full picture allows us to make better decisions, not just as scientists, but as human beings. Fetal bovine serum helped build modern biology. It keeps cells alive, supports research, and has contributed to discoveries that improve lives around the world. But it also reminds us that scientific progress is rarely simple. The future of biology may involve better alternatives, new technologies, and entirely new ways of growing cells. The question isn't whether science should continue advancing. The question is how. Because the future isn't written by technology alone. It's written by the choices we make, together. Thanks for learning with me today. If you learned something new, please like and subscribe for more science videos. We've got lots more weird, fascinating, and surprising science facts to explore. See you soon. Bye.